Welcome back. So, last time we were discussing all of the different anatomical components of the heart, and now we're going to move into the physiology of it. We're going to look at how it functions, which is definitely going to be a little bit of a bigger topic than just the structures. And that's just kind of how 252 is going to go, where the structures are the most basic component, and then the physiology is really where we're going to spend most of our time. So the first thing we need to do is we need to talk about cardiac muscle. So you know that this is one of our three types of muscle, and you should remember that it is striated in appearance, but it is involuntary. So it has one characteristic similar to skeletal and one characteristic similar to smooth muscle. So cardiac muscle fibers are also known as cardiomyocytes. They are striated and they are going to contract via sliding filaments in a very similar way as skeletal muscle. That is where your similarities between the two end. In terms of the structure, remember that skeletal muscles are very long, so the fiber is the length of the muscle. In cardiac muscle, we have short branched cells. So the cell goes from here to here, but there's a branch of it coming over to here as well. So we're going to have to connect them with what are called intercalated discs. They show up as these very dark bands in histology. This is going to be what anchors these very short cells together. Picture a skeletal muscle, biceps brachii. It has attachments in your shoulder and attachments in your forearm. And so when they begin to shorten and you're going through your muscle contraction in your sliding filaments, you're pulling on something very, very sturdy. You're pulling on bones, and it's one long cell throughout there. Now imagine if it was a whole bunch of individual cells back to back to back to back to back going from your shoulder to your forearm. You would need something really, really heavy duty holding those cells together so that when they contract, they don't just fall apart. That's where the intercalated discs are going to come in. So what we have here is going to be several different types of cell junctions. So we've got desmosomes. So these are going to connect the intermediate filaments in neighboring cells. We have the fascia adherens, which is connecting neighboring actin filaments. And then we also have gap junctions. So these two, the desmosomes and the fascia adherens, are going to hold the cells together. Gap junctions are going to allow the signals to pass very quickly from one cell to another. So very important structure in the intercalated discs here. Now the pattern of kind of the egg crate pattern, if you will, is going to increase surface area so that you have more area to hold these together. So you have more surface area for the cell junctions to occur on. There is somewhere in the neighborhood of 1 to 2 nuclei per cell. So a lot of them only have one, but you will find a decent number of them that have multiple nuclei in them. The nucleus is usually surrounded in glycogen, which is going to be a storage for our glucose here. We have a ton of mitochondria, so these things are just packed full of mitochondria. We do have a sarcoplasmic reticulum, but it is going to be a little bit less involved. It doesn't have the terminal cisterna and things like that, and we tend to see larger T tubules here. So structurally, really the major similarity is the striations and the fact that it's going to contract via sliding filament theory. Everything else is pretty much different. Functionally, they are completely different. So skeletal muscles are going to be stimulated via our motor neurons, which we talked about in 251. Cardiac muscle is going to use what are called autorhythmic cells. So we're not using a neuron to stimulate these muscles. The contraction is different. So a skeletal muscle is going to contract in a motor unit. So remember that that's a motor neuron and all the fibers it innervates. So when that neuron fires, those particular fibers will respond. And so you can have a muscle that has multiple motor units and all of them would have to fire in order for all of the fibers to contract. Cardiac muscle contracts as a whole. In other words, once you begin this process, all of the cardiomyocytes are going to respond as if they were one single cell. It's called a functional syncytium. So it means that you have multiple things that are functioning as one single unit. There is a big difference in the refractory period. So in skeletal muscle, that absolute refractory period is about 1 to 2 milliseconds. In cardiac muscle, it's 200 milliseconds. If you remember, when we were talking about skeletal muscle, we said that one of the ways to increase tension in the muscle was to speed up the frequency of stimulation. And we said if you went far enough, you could actually get to tetany, which was a sustained contraction. We don't want your heart to get to tetany, and so we have this very long, drawn-out refractory period of 200 milliseconds. It's going to prevent your heart from seizing and also allow us to have enough time to undergo what we need to do in the cardiac cycle. Lastly, the energy requirements are also different. Skeletal muscles obviously can use aerobic respiration, but they also have the option of anaerobic respiration as needed. They can do lactic acid fermentation. Cardiac muscle has an absolute requirement for oxygen. There is no ability to undergo anaerobic respiration. When you cut off the oxygen supply from cardiac muscle, the cells will die very quickly, and they die at an alarming rate. You're talking about somewhere in the neighborhood of 500 cells per second if you have a blockage in your coronary arteries. So that's why we are so concerned and why we want to get people into the cardiac cath lab or something like that as fast as possible to intervene in those situations, because every second we are losing heart muscle. And remember that cardiac muscle does not divide. It will not regenerate itself. So let's talk about the electrical events in the heart. The heart is going to depolarize and contract without the nervous system stimulating it. Now, we should remember that the nervous system affects the heart. You did an entire chapter or unit on the autonomic nervous system, and you know that the sympathetic nervous system increases it and the parasympathetic decreases it. And so you know that the nervous system affects the heart, but we can take a heart out of somebody, and as long as we give it ATP, then it will beat on its own. It does not need to be connected to the nervous system to undergo the cardiac cycle. That is due to those gap junctions and the cardiac conduction system. So this is a series of interconnected autorhythmic cells. So these are non-contractile. They do not contract. Their job is to set the pace for the cardiac cycle. Therefore, we call them pacemakers. So they are going to have a non-stable resting membrane potential. So in a normal cell, you come down to resting membrane potential and you stay there until you're stimulated. In these autorhythmic cells in the heart, there is a very slow influx of sodium due to the membrane structure that is going to, as soon as we get down to resting membrane potential, we begin to depolarize. And when we hit threshold, we have an action potential. And then we come back down to resting and we go immediately into depolarizing. And that's without the nervous system. So there's nothing opening up a channel. It's just the fact that the membranes are leaky. So the portion of this that exists between resting and threshold is called a pacemaker potential. So unlike our skeletal muscles, we don't require acetylcholine. We don't require anything for this to happen. The leaky membranes are going to allow this sodium to very slowly, and by slowly, we're talking about like 0.4 seconds here. So that's a relative term. It's going to very slowly leak in and drive us closer to threshold, and at that point we have our action potential. So this conduction system starts up in what's called the SA node or the sinoatrial node. This is what is known as the pacemaker of your heart. The signal is going to travel through the very thin atrial walls and through that very thin interatrial septum. And so it's going to cross over to the left very, very easily, and you're going to depolarize the atria.
Now, the way we want the heart to contract is we want the atria to contract from the top down, but we want the ventricles to contract from the bottom up. And if you studied your blood flow, you understand why. If I want the atria to contract in this direction and the ventricles to go and contract in the opposite direction, it means I have to send the signal in those directions. So the contraction follows the signal. So the atria, we start up here and the signal moves downward, and it's going to allow the depolarization and then contraction of the atria going down. But if that signal just kept going straight down, the ventricles would also contract in that direction, and that would not work. So we have this layer running right through here of dense connective tissue called the cardiac skeleton. And this is where it comes into play when we were talking about the fact that it's an insulator. So the signal can't get through that dense network of connective tissue running through the area about right here. And so it can't go from the atria to the ventricle directly. So there's one spot that is conductive, and that is the AV node. And it's kind of functioning similarly to almost like a lightning rod right here. All of this electrical activity is going to get drawn to this one spot that can conduct it. So the AV node is where all of this electrical activity is going to meet up. And then the signal will pause at the AV node. Once it fires, it's going to go down the bundle into the bundle branches, and it's going to travel up into this subendocardial network. And once it starts traveling through here, it is actually going so very fast, okay? It's traveling at 4 meters a second through here, that by the time the muscle can actually depolarize following all of this, it's going to be following the upward trajectory of the signal, not this downward piece right here. So it's coming down the interventricular septum, but it's going so fast that the muscle begins to respond somewhere in here, and so the response is going to follow the signal, and the response is going to go from the bottom up. So when we talk about the rhythms that are created by the SA node, the AV node, anything that is being generated in a normal rhythm by the SA node is called sinus rhythm. So this is what we would term typical. An arrhythmia is anything that isn't a typical rhythm. So it means that there is something abnormal about it. Some of them are fairly common. Tachycardia and bradycardia, for example. Tachycardia is when our heart rate is higher than normal. I mean, if we all decide to go exercise, we're going to become tachycardic. Bradycardia occurs when our heart rate is lower than normal. This happens to all of us when we go to sleep. Okay, so there are some, abnormal rates that are fairly common. Now, not to say that either of these cannot be pathologic in certain situations, but they are also normal situations that we would see these. Sometimes an arrhythmia is driven by what's called an ectopic focus. So here is basically where something other than the SA node is driving the rhythm. I'll give you a couple examples of this. One is called junctional rhythm, and this occurs when the SA node is damaged and the AV node takes over. When the AV node takes over, you're still going to get contraction of your ventricles, but it's going to be at a much slower rate than normal. This isn't necessarily something that's going to cause you to drop dead immediately, but over time, it's really going to tax your system. Another example is heart block. This occurs when there is a true blockage at the AV node, meaning that no signal is entering into the ventricles. And this is a big, significant, major problem. There are different degrees of this. By the time you get to 3rd degree heart block, you will drop dead. The ventricles will contract on their own anywhere between 10 to 30 times a minute, but it will no longer be coordinated with the atria. And so this is pathologic and possibly fatal. We do know that the basic rhythm can be modified as needed via the sympathetic and parasympathetic systems. So when we're resting and digesting, parasympathetic can decrease our heart rate and make us bradycardic. When we're exercising, the sympathetic can kick in and make us tachycardic. Okay, so the nervous system certainly communicates with the heart by interacting directly with the SA node and the AV node, but remember that we don't need this. We can set our basic rhythm without the nervous system's involvement. All right, so let's track this electricity through the heart. So you need to remember, first of all, what depolarization and repolarization are. So depolarization is the flow of sodium in, repolarization is the flow of potassium out. And so this is really it's an electrical current is what we're looking at. And it's important to understand that these are electrical events that we are watching, not physical events. So yes, depolarization leads to contraction, but when we see this right here, this P wave, that is not indicating atrial contraction. It's indicating atrial depolarization. The contraction will follow that. The electrical events lead to the physical events. Okay, so starting up in our SA node, okay, well, we start here with no electrical activity. The first thing we're going to come to is the P wave. And this is representing the atria depolarizing. You can see the red, that's a wave of depolarization going through the atria. Once the atria have depolarized, I said we were going to pause at the AV node, and so we come back down to no activity right here. This is called the PQ interval. When you get into the QR, you can see QRS, there's the completion of it. The QRS is going to represent ventricular depolarization. So it comes down the bundle in the interventricular septum and then hooks up the apex and starts going all through the muscle there. So the QRS, it's completed over here, is ventricular depolarization. Then we go back to a period, the ST segment, where we are not doing anything. We're going back to zero electrical activity. And then we come across our last component of the ECG, which is the T wave, which represents ventricular repolarization. And then once that is complete, we're back to no electrical activity. So our P wave is indicating atrial depolarization. The QRS complex is ventricular depolarization. The T wave is ventricular repolarization. Now, the atria do repolarize as well. However, it is masked by the QRS because the ventricular depolarization is a much bigger event. So we don't actually register it on the ECG, but it does happen. Okay, so the cardiac cycle is really a pretty significant big component. It is what we've been working towards, and you are going to need to be able to interpret and understand this diagram. It's called Wigger's diagram, after the guy who made it. And when you get to this point, understanding this is going to require that you understand the anatomy of the heart, that you understand how how valves work, which ones open with increased pressure, which ones close with increased pressure. It's going to require that you understand the flow of blood through the heart and the conduction system. So really, this is where we wrap up all of those different components that we've talked about into the function of the heart. So you need to really spend some time here. You're going to have a complete lab over this as well. So this will be the subject, this diagram will be the subject of your cardiophysiology lab. So you're going to be walking through this with me right now and then also again in lab. Okay, so let's start by what is actually in this diagram. First of all, we have heart sounds. Heart sounds occur when valves close, not when they open. So let's just remember that AV valves close when the ventricle fills with blood and the pressure increases and causes them to close, while semilunar valves are going to fall close when the pressure beneath them decreases. So when we see heart sounds, we should see some indications elsewhere in the graph that pressures are changing. The ECG we just talked about. So we know we have our atrial depolarization, our ventricular depolarization, and then our ventricular repolarization. So somewhere in the graph, when we see these, we should see indications that following the P wave, the atria contract. Following the QRS, the ventricles contract. Okay, but remember that this line is not showing you contraction. It is showing you electrical events. This is ventricular volume. So we're going to track the volume of blood in the ventricles through the cardiac cycle. So we can see that there's a period where it goes up and then down and then back up and down. And then this is pressure. Okay, so we're going to get to this pressure one in a minute. Let's just talk about volume for a second. There's actually quite a bit of information to be found simply by looking at volume and what's going on with valves. So in this portion of the graph here, ventricular volume is increasing. So if ventricular volume is increasing, the blood is coming from an atria into the ventricle. That's the only way for blood to get into a ventricle is from an atria. So there is a valve between them that must be open. So your if-then statement regarding increasing ventricular volume is if ventricular volume is increasing, then AV valves are open. There's no way for volume to increase and have the AV valves closed. So anytime you're seeing increases in volume, you know AV valves are open. If you look right here, volume decreases. If volume is going to decrease, then it means that the blood is being ejected from the ventricle. And to be ejected from the ventricle, it's got to go through a semilunar valve. So your if-then statement here is if ventricular volume is decreasing, then semilunar valves are open. So just by tracking whether volume is increasing or decreasing, we can tell when AV valves are open and when semilunar valves are open. We've got a couple areas here, right here in this kind of yellow stripe and right here in this purple stripe where volume doesn't change at all. The only way, go back to your if-then statements, the only way for volume not to change at all is if all four valves are closed. These are what are called isovolumetric phases, meaning that the volume is not changing in the ventricles because all four valves are closed. Because we know if the AV valves are open, then volume is going to increase. And if the semilunar valves are open, volume is going to decrease. So the only way for volume to stay the same is all four valves are closed. Now, there is no point where all four valves are open. That would be a problem in terms of the flow of blood, but also, let's go back to what we've already talked about, increasing ventricular pressure closes AV valves and opens semilunar valves. Therefore, there's no time in which all four valves could be open at the same time. They open under opposite conditions. Okay, so really just by tracking ventricular volume, we know AV valves are open, all four valves are closed, semilunar valves are open, all four valves are closed, AV valves are open, all four valves are closed, semilunar valves are open, and you just keep going. So there's a lot of information just by looking at volume. All right, now let's add on the, let's come up here and talk about pressure. Let's go over what we're looking at here. So there's 3 lines in pressure. Now, everything in this chart is applicable to both sides of the heart except the pressure. The pressure on the left side of the heart is far greater than the right. The right ventricle is pumping into the lungs, which is incredibly delicate tissue and cannot withstand the force that the aorta can. So this piece right here is specific to the left side, and you do need to reference that. So we have a line down here, this red line down here, that stays at a very low pressure all the time. So, between like 0 and 20-ish maybe. We have a line up here that stays at a very high pressure. So it drops down to about 80 and then goes up to about 120 and then eventually drops down to about 80 and then 120. And then we have a black line that goes between the two of them. It starts at low pressure and then ends up at this very high pressure and then comes back down to low pressure. So the three sides of the three parts of the left side of the heart that are being represented here are the left atria with very low pressure. That should make sense. Remember that we don't have a lot of muscle, so we're not going to be able to create a lot of force. The aorta, which should stay at a very high pressure because it's a blood vessel, and so what you're tracking here is actually blood pressure. And we know if blood pressure is ever around 0, we've died, so that's not good. And then the ventricle is the black line. So the left ventricle has the ability to be at very low pressure and then generate huge amounts of pressure and then come back down to low. So left atrium is the red, aortic is the upper high pressure red line, and then the black line is the left ventricle. So we're going to start right here with this, let me see. Yeah, we're going to start right here with this blue line. So right there, okay? This is the start of a cardiac cycle. And at this point, where we're going to start, the heart is in what's called a diastolic phase. Diastole means relaxation, systole means contraction. And generally speaking, when you see this up here, it is referring to the ventricle. But right here, we are in this blue stripe right here, this darker blue stripe in what's called phase 1A. Everything is diastolic. Everything is relaxed. We know that the AV valves are open because ventricular volume is increasing. And we can see that the pressure in the left atrium and the left ventricle is fairly low because they are very big and relaxed. And they are very similar because the valve is open between them. So pressure is going to equalize between the two chambers if the valve is open. So that's where we are starting. Now, this little S3, this is a possible third heart sound. We're going to ignore it, okay? It's not constantly heard, and it doesn't have to do with valves closing. It has to do with this flow of blood into the ventricle, but you don't generally hear it, so we're not even going to worry about it, okay? So just ignore it. You can make a big X through it if you want to, the S3. So this is where we are. The ventricle and the atria, everything is relaxed, the AV valve is open, blood is flowing in, pressures are equalizing between the two chambers. Then we are going to come across the P wave. So this is our first thing. So we're in this blue area, everything's going. We've got equal pressures, volumes increasing, and then we come to our first part of our ECG, the electrical event. So the SA node is going to fire, and we're going to see the P wave. Following the P wave, if we come up to the pressure, we see a little blip. Not a lot, okay? This isn't a huge change in pressure, but it's representing atrial contraction. So the atria are going to contract and push blood into the ventricles. And because the atria are contracting, you get this little blip of blood. So what atrial contraction represents is kind of a topping off of the ventricles. So we're putting as much blood into the ventricles as we can with atrial systole. Now, it's not a lot. It's a contraction that just kind of goes bleep. Okay, that's about it. And remember, it's pushing with gravity. And so we're really just topping off the ventricle, making sure that we get enough blood in there to change the pressure in the ventricle and close the AV valve. So at this point, when we have filled the ventricle, the AV valve is going to be forced shut because of that volume in the ventricle. Now, this period right here was where the signal was pausing at the AV node in order to make sure that the ventricle gets fully filled with blood before contraction. So then we head into the QRS, And so that's the electrical component telling the ventricle getting ready to contract. And at the point where the AV valve closes, we hear our first heart sound. So the first heart sound is going to represent the AV valves closing. So the right and the left. So the bicuspid and the tricuspid are closing, and we're going to hear that heart sound while the QRS is occurring. Now, once the AV valve closes, we are going to enter into this phase right here, which is called isovolumetric contraction. The AV valve has closed, but the ventricle has not yet generated enough pressure to open the semilunar valve. So this is a point where all 4 valves are closed and the volume stays the same. This volume right here, the highest possible volume, is called the end diastolic volume, or the EDV. The QRS is going to tell the ventricle to contract, and it is going to begin to contract, and we can see that because this line increases. The ventricle is increasing in pressure. It is contracting from the bottom up, and it is twisting while it shortens and contracts. So you're getting kind of a double generation of pressure inside the chamber, because not only are you shortening, you're twisting. So we're going to see the ventricle gain pressure. And remember where the semilunar valves are. They are at the top of the ventricles, and the ventricles are going to have to force that valve to open by pushing the blood, pressurizing the blood, and pushing the blood up against that valve and opening it. Remember we said that this is like a door in the ceiling, and so we have to push up on it. So it's going to take us, a minute to push up and hit enough force to actually open it. So what's happening during this time, during this isovolumetric contraction, is the ventricle is contracting, but we haven't yet opened the semilunar valve. At this point where the ventricle, the pressure exceeds the aortic pressure, the pressure that's pushing down on that valve, and you generate enough and you actually cross, then at that point, volume begins to go down because the aortic valve is going to open, and we see this with the ventricular volume decreasing. Now, the contraction isn't over, so the ventricle is going to increase in pressure until it reaches its highest pressure, because cardiac muscle responds completely and fully every time. There is no graded response in cardiac muscle. You tell it to contract, it's going to go through a complete and full contraction every single time. So it's going to reach its peak here. And again, once the aortic valve opens, the pressures in the ventricle and the aorta are going to be the same, because pressure is going to equalize. So whenever you see the lines running parallel to each other, it means that the valve between those two things is open. If they're not running parallel, like here, the aorta and the left ventricle, that means that the valve has not opened. Okay. So we're going to go through a full contraction. So we get to our peak contraction up here at about 120 millimeters of mercury. And then because the contraction is complete, the heart is going to begin to relax. That's all it can do. It's already completely contracted. There's no more it can give. It's 100%. And then it begins to relax, and it begins to shorten and unwind. So at this point, the pressure is beginning to drop because the chamber is getting bigger. And remember, what was holding this aortic valve open, this semilunar valve open, was the blood that was pushing up through it. So eventually, as the pressure drops, the valve is not going to be able to be held open anymore, and we're going to get to this point where ventricular pressure drops below aortic pressure and the aortic valve closes. At this point, we come down here and we see our second heart sound. So your second heart sound is going to be represented by what's making it is your semilunar valves closing. So the pulmonary and the aortic valves are closing, and you hear your second heart sound. Generally, we say the first one is called LUP and the second one is DUP. So first one is AV valves, second one is semilunar valves closing. Now, the aortic valve is closed, but look what's happening. We're in another isovolumetric phase because the aortic valve has closed, but ventricular pressure is still high. So the AV valve has not opened yet. So this all goes back to the forces that open and close those valves. If you don't have a good understanding of that, you need to go back to that and then come back to this. So the pressure in the ventricle beneath the AV valve is still fairly high, and so we are going through this isovolumetric relaxation phase. And eventually, T wave, so we're repolarizing, eventually we're going to drop below the atrial pressure, and at that point the AV valve is going to fall open, and we begin our next cycle by filling up the ventricle again. So now we've circled back around to our blue phases. So based on what's going on in the P wave and the QRS, you're going to see what's happening here. You need to walk yourself through this. And honestly, one of the best ways to do this is to sit down and draw this out and make sure that you understand what's going on in these pressures, because this is usually the part that is going to cause people the biggest problems. And to understand this, you have to understand, first of all, the anatomy of the heart, because you need to know what valve is between the left atrium and the left ventricle that closes. You need to know what valve is here. Okay, you need to understand the flow. You need to understand that AV valves would close, and then an aortic would open, then the aortic would close, and then an AV valve would open. You have to understand the pressures involved. So you really have to understand the valves and the blood flow to get what's going on in this pressure reading. Now, once we begin to, once this valve opens, okay, then we start gaining blood. When we are at this lowest volume right here, this is called the end systolic volume, or the ESV, okay? So the highest volume is right before the ventricle goes into systole. It's at the end of ventricular diastole. So it's the highest volume because we spent all this time filling the blood or filling the ventricle passively, and then we added an atrial contraction to get us up to EDV. Once we have contracted the ventricle, we're going to lose blood, and we're going to end up down here at our lowest possible volume that occurs at the end of ventricular systole, so it's the end systolic volume. Notice it is not 0. So you're going to, go anywhere. You're going to lose somewhere in the neighborhood of 70 mils of blood here, but you can never actually get rid of all the blood because the ventricle is pushing blood straight up. Picture that you have a bucket, and it's your job to empty this bucket that's full of water. The ventricles, the only way to empty the bucket is to throw the water straight up in the air. That water is going to fall back into the bucket, so you're never going to be able to empty them. Okay, so you need to spend time here. This is definitely not something that people generally are going to get the very first time they hear it. That's why you're going to have a complete lab that is going to cover this. You're going to go over it multiple times in this video, and then you will be going over it in lab as well. Really, you have to understand all the components that I talked about from the first heart video and everything leading up to this chart before you can really understand this. So you really need to spend some time here. I will put some questions on your homework so that you can practice on this. It is a really important concept, and it's definitely something that requires some time. All right, so let's talk about cardiac output for a second. Cardiac output is stroke volume times heart rate. So let me go back here for a second. Stroke volume is represented right here. It's EDV minus ESV. So it is the volume of blood that is ejected per ventricular contraction. Stroke volume. So cardiac output is the volume of blood pumped out by the ventricles per minute. So you have to take stroke volume, which is usually about 70 mils, and multiply it times heart rate, which is how many times do we do this per minute, and you're going to end up with a cardiac output, which is somewhere in the neighborhood of 5 or so liters a minute. But if you remember back to blood, that can feasibly be your entire blood volume. It does not take long to cycle your blood through your body. So per minute, you're basically you're cycling your blood. Okay, so let's talk about what cardiac output is. Cardiac output is the product of stroke volume and heart rate. So it's the volume of blood pumped by the ventricles each minute. And you need to take stroke volume, which is usually about 70 mils. Stroke volume is the difference between EDV and ESV. So it's represented on that chart we just looked at. And then you need to multiply it by how many times you do this per minute, and you're going to end up with somewhere in the neighborhood of 5 or so liters of blood, which, if you remember, is feasibly your entire blood volume. So you cycle your entire blood volume through your heart per minute, essentially. So cardiac output, stroke volume, heart rate, these are going to be significant players in blood pressure, which is where we're headed in our next video. But let's talk about stroke volume and heart rate for a little bit. Let's talk about how we regulate them. Okay, so we're going to talk both of these. We're going to start with stroke volume. So if you think about stroke volume, you want to think about, okay, how much blood is the ventricle ejecting per contraction? And that's all going to depend on what's going on in the ventricle and how well it contracts. So one of the first things that's going to affect stroke volume is contractility, which is related to the availability of calcium. Calcium binds to troponin, gets everything going. If you don't have calcium, you're going to have a hard time doing this. Okay, so stroke volume will suffer in the absence of calcium. Then you have two concepts that tend to be somewhat hard for students to understand, which are called preload and afterload. Preload has to do with how much blood is in the ventricle at the end of diastole. Essentially, preload is related to EDV, end diastolic volume. The idea of preload is that we need to stretch the ventricle before it contracts. Sarcomeres function best and generate the highest pressure, the highest force, if they are slightly stretched before you contract. Now, there's no way to, just grab your heart and stretch it from the outside. You have to stretch it from the inside. That's why that atrial contraction is important, because it tops off the ventricles and allows you to get to a good high EDV, which allows preload to occur. It allows you to stretch the ventricle before contracting. So anything that affects how much blood is coming back into the ventricle, so like poor venous return or something along those lines, any of those, they would negatively affect preload, which would then negatively affect stroke volume. Afterload is the amount of pressure that is being applied to the aortic valve from the blood sitting on top of it. If you picture the aortic arch and you picture the ventricle contracting, and it's going to be pushing on that blood, and that blood's going to go out into the aortic arch, but then you get to that peak of the contraction, and then the ventricle begins to relax. And so there's going to be a closure of that semilunar valve, and whatever blood didn't make the arch, gravity is going to pull it back down, and it's going to sit on top of the valve. So it's going to sit there on top of the valve, okay? As it sits there on top of the valve, it is applying a pressure that the ventricle will have to overcome the next cycle. That is afterload. It's the resistance that the ventricle is encountering when it tries to open the semilunar valves. Whenever stroke volume goes down or force of contraction goes down, afterload goes up, which will then further cause problems with stroke volume. You got to spend some time with these, okay? The other half of this equation is heart rate. The autonomic nervous system is a primary regulator of heart rate. We know this. We've got sympathetic and parasympathetic innervation, so we can speed it up or slow it down as needed. The other component that's going to come into play are different chemicals. We're going to talk about different hormones and different ions that can affect how well your heart is going to undergo this rate. Okay, so both sides of our cardiac output equation have multiple components that lead into them. And when we begin to look at blood pressure, we're just going to go over all of this again. So we're going to see this in play and really begin to put together the pathways and the mechanisms.